Okay, so today we will study about endomembrane system. So the last two classes we have studied about, the first class we studied about a typical prokaryotic cell structure. Then the next class we studied about cell wall and cell membrane. Today we are going to study the structure inside the cell. That is, in the cytoplasm there are various types of membrane bound structures in a eukaryotic cell. From that we are going to study 4 of them, which are always explained as endomembrane system, because it works as a system. It works in a coordinated way, in an interconnected way. So we call it as endomembrane system. So all the members of this endomembrane system, they are membranous structures or membrane bound structures, and these membranes are made up of phospholipid bilayer. So the same as in the cell membrane, there we say about proteins also, but here we just mentioned about the lipid. So phospholipids are there, they form a bilayer forming the membrane. Now, which are the cell organelles coming under endomembrane system? First one is endoplasmic reticulum, then the Golgi complex, otherwise called as Golgi apparatus, then there are vacuoles and lysosomes. So first we will study endoplasmic reticulum and Golgi complex. Now, in lower classes you have learned about endoplasmic reticulum. So these 4 that is endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. They are interconnected, means they work together and do various functions in the cell. So in case of endoplasmic reticulum, its main, what is endoplasmic reticulum? They consist of various tubular structures. So it is formed of membranous tubes, cylindrical structures, or we can say it as membranous folds in the cytoplasm, which divide the cytoplasm into various compartments. This compartments or compartmentalization is very important for the effective functioning of the cell. So that is the main function done by endoplasmic reticulum. So they compartmentalize the cytoplasm into various regions. So based on it, there are 2 types of compartments. One is called the luminal compartment. This is the part inside the endoplasmic reticulum. So it is seen in continuation with the perinuclear space of the nucleus. So nucleus is covered by a double membrane. Between the 2 membranes there is a space called as a perinuclear space. So this endoplasmic reticulum is seen in continuation with the perinuclear space. Then extraluminal compartments are regions in the cytoplasm, that is outside the endoplasmic reticulum. Now, based on whether the ribosomes are present on the surface of endoplasmic reticulum or not, there are 2 types of endoplasmic reticulum. There is rough endoplasmic reticulum having ribosomes on its surface, and a smooth endoplasmic reticulum which are devoid of any ribosomes attached to its surface. Now, rough endoplasmic reticulum, since it is having ribosomes on its surface, ribosomes are called as the protein factories of the cell. So since the ribosomes are present, they are mainly concerned with protein synthesis and their export or the transport. Smooth endoplasmic reticulum is mainly concerned with lipid synthesis, like cholesterol and all. So this lipid synthesis and from cholesterol various different types of hormones and all are formed. Like steroid hormones are examples for it. Like estrogen, then androgens. All these are steroid hormones, means hormones which are derived from cholesterol. So lipid synthesis is the main function of smooth endoplasmic reticulum. They also help in detoxification. Okay, so they are usually in human body, they are present in gonads. Smooth gonad cells, there are a large number of smooth endoplasmic reticulum. The same way liver cells have large number of smooth endoplasmic reticulum because it helps detoxification. So liver is the organ which does detoxification. So then transport is a common function for both the type of endoplasmic reticulum. Now coming to Golgi complex. It's also called Golgi, simply Golgi, or Golgi apparatus, or Golgi bodies, or Golgi complex. Now, this was first discovered by the person called as Camillo Golgi in the year 1898. So since he discovered it, it is named after him. Now, he observed it as a densely stained tubular structures near to the nucleus. Okay. Now, it consists of, and it's called as a complex because it consists of 2 types of structures, vesicles and flattened sacs. In the vesicles എന്നുള്ള term എപ്പോഴും ബയോളജിയിൽ വന്നാലും ഉദ്ദേശിക്കുന്നത് these are membrane, fluid filled membrane bound sacs are called as vesicles. So vesicles in കാണാം അതുമായിട്ട് so we can also see membranous folds, or we can say flattened sacs of membrane. So these flattened sacs are called as cisternae. The cisternae are arranged one above the other. That is called that they are stacked parallelly one above the other. In cisternae there are two types are there. One with convex surface, we call it as a cyst type. Cyst is a cyst phase in the variety. Cyst phase is the formation phase, or this is the region where things are newly formed. Then trans phase lana the exit phase the forms of cyst in the translate polar materials. So there is a cyst phase and there is a trans phase. And in various stacks of this Golgi bodies, there are different types of enzymes. So each compartment of the Golgi body is having various enzymes by which the proteins which are transported from endoplasmic reticulum. So endoplasmic reticulum can transport things between one cell and the next cell. It can just transport, but it should be transported in the form of vesicles. Okay, it will first transport to the cyst part of the Golgi apparatus. From there, it is traveling towards the trans region, and during that time it gets modified by the enzymes present inside the Golgi body, and finally it forms a vesicle and is released out. So like that, that's the function of Golgi complex. That's why we call it as the packaging centers of the cell. So the main function of Golgi complex is packaging. Plus, it also does secretion of certain substances like glycolipids and glycoproteins. So that is the function of Golgi complex.